सर किसको बनाना है? सब
Okay. Yeah, good. It's on. Yep. So after that lecture, followed by a discussion. After lunch, you come back here, and we have 3 practicals lined up. Yours, Victor, Lisa, Guillermo, they will be handling these practicals. And the last practical is the observation of those leaves, the concentration of this is what we have done. So with that, we will close today's activities, whatever we have. So what we are about to do with the students' presentation. I'm sure the students' presentation are ready, right? So try to submit by this evening. I'll give you at least all of them. And that will take it up later after this, after the lecture. I think I will call upon those parameters to handle the first lecture of this time. Okay, thank you, Kumar. Good morning. The mic is on, right? Yeah. Good. Apparently there was some miscommunication with the travel agent from AAR, so which means that we already fly in the night today and not, so we're going to be here a day less, which I think we regret, but it's, I mean, it is what it is. We can't change that anymore. Which means that for the literature presentations tomorrow, I already prepared an online Zoom link, so we will have the presentations online. So when we arrive in the Netherlands, we go home by train, and then we'll get online again to meet you again. It does mean that in terms of the 4.5-hour time difference, that the program will change a bit later for you. Anyway, it also gives you a bit more time to prepare for the presentation, okay? Good, so last Friday we joined the Convocation Day, which beautiful ceremony. So I also learned that day that some students here, they are, you know, actually talented singers as well. So that was nice to see. Good, so for today's program, initially I had planned to give 2 lectures on the functions, but because of the keynote lecture today and the program, what I did yesterday, I merged the 2 lectures on functions, I merged them into one presentation, which is going to be a bit longer than usual. So I'll walk you through. So we talked, there was an introduction, you remember, there was 2 lectures on assembly, and this will be 2 lectures in one on functions or functional analysis of plant microbiomes. And again, I want to bring this slide back again. So you remember we had 2, yeah, maybe you could. Yeah, is it better visible now? So you remember we had 2 types of, yeah, you can study microbiomes in roughly two manners. One is when you look at the microbiome as a phenotype. So what is the impact of environmental conditions, of the plant genotype, on plant development, on the microbiome composition and functions? That is one way to look at it. But you can also study the microbiome in the reverse. So what is the plant genotype associated with the microbiome, and which members of the microbiome contribute to that trait, and which functions within the microbiome contribute to that trait. Right? So those are 2 things, and we call this MAPS, microbiome-associated phenotypes. And these now will be mixed a bit into. Now, this is the most important slide of the lecture. This is a flow that was proposed by Julia Forhold in 2017, how to study functions of the microbiome. So normally you start with plant and microbiome as, as a collection, so they are together here. And to study functions in the microbiome or in the plant related to microbiome assembly or, for example, microbiome-associated plant phenotypes, you disassemble the two. Yeah, so basically you separate microbes from the plant. And it all depends on what you're interested in. If you're interested in archaea or viruses or bacteria or eukarya, usually in this stage here, you start to establish collections of, the group of organisms that you're interested in the microbiome. And then the idea, and that's the second stage here, it's called a reconstitution. And reconstitution is you start to try to build the microbiome that is here for a particular phenotype that you're interested in. And I know there's people here that work on synthetic communities, right? So to design that, I will come back to that in a minute. So you start to reconstitute the microbiome, and then there are two types of readouts. You can look at the microbial phenotype, so you can look at how they establish on the plant surfaces, how they express specific genes on plant surfaces, and you can look at the host phenotype. So it's basically the phenotype that is associated with, if you apply a microbe, what is the readout in terms of phenotype. Here there's a number of things also related to the microbiome, which some of them we talked about. So there are a number of terms here: resistance, resilience, contingency, invasiveness, and priority effects. We talked about priority effects, right? The time of arrival when a microbe gets onto the plant. Invasiveness is when you, for example, apply a microbe or a probiotic. How well is it to invade an existing microbiome there, so to establish itself? Contingency has to do with the predictability of microbiome assembly across different environments. And then two terms, resistance and resilience. Resistance of a microbiome means that if there is a perturbation, usually the microbiome composition does not change. So it's resistant to that perturbation. Resilience is a bit different. So then you do get, a change in composition or in functions, but resilience refers to how fast the microbiome then comes back to its original composition, right? So those are the two. So again, and especially disassembly and reconstitution will come back later on in the lecture. So first I want to go through a couple of microbiome-associated phenotypes. Some of them are well known to you: plant growth, plant development, plant quality. I will go also in here. And then, of course, stress tolerance, either to abiotic stress or to biotic stress. Okay? Let's start with the usual suspects here. Biological nitrogen fixation with, of course, the rhizobia here. Very well-known system, although there are still many aspects to learn here. Some people here, they talked about not only the rhizobium, but also other microbes associated with the nodule, right? So that's still a nice field of research. Yeah, there's not much to say here. And then, of course, the other ones, so the ectomycorrhizal fungi or the endomycorrhizal fungi, basically the symbionts. And then here you have, a number of plant hormones like auxins, gibberellins, and ethylene, which, of course, impact on plant development as well. And then here, I think we also talked about that, about the chemistry of the rhizosphere environment where plants release organic acids to solubilize, micronutrients. And also on the microbe side, they have systems to solubilize these micronutrients for growth, right? So this is sort of like baseline. But it's worthwhile to look into this review paper. There's a lot of other beautiful cartoons that depict these types of processes. Okay, then, so these are sort of like the usual suspects. And then I want to show you, I always show this picture, and I will show it also in my keynote lecture, so you will have it twice today, about what microbes can, how they impact on plant growth. And this is a so-called blind date experiment, which was done by a PhD student in my group. He's a biochemist, so he had not much knowledge on microbiology as such, and he was interested, okay, what can microbes actually do for plants? And at that time, he was also looking for a girlfriend, so that's why blind date came up, right? But what he did is we took 3 random bacteria from the freezer, and he took 3 plant species, and he paired the 3, right? And this was the result. You can also do microbiomes if you want, but he did a microbe, 1 microbe at a time. So here you have the plant species, Arabidopsis, which is a model plant. Then you have a medicinal plant, Artemisia, which is the producer of artemisinin, which is an anti-malarial drug, right? And then here you have a food crop, broccoli. So these are the plants growing on an agar plate in absence of the microbe. So this is basically your sterile control. And then what Jae-seung did is he first germinated the seedlings of all these plant species, and then once there was a root tip, he inoculated 1 microbe at the root tip. You can also do this with more complex communities like a microbiome. And then what he did is he let these plants grow for another 2 weeks, and then these were the phenotypes that he observed. So here, this phenotype, there doesn't seem to be much, changing, right? Then you have a very typical phenotype you see with microbes, that you get much more lateral root formation, and the primary root is shorter, but you get much more branching of the root system. Then this phenotype, you can see this plant is totally stressed. So this is the phenotype that we call in my lab, too much love can kill you, okay? So you can see also in terms of chemistry, a number of changes. I will come back to that later. And then, of course, this one, this is just an amazing phenotype where, the microbe, creates, I think it was about a 6-fold increase in biomass. And you see that also the root system is totally changed. So I think all these sort of scenarios that you can imagine with a microbe interacting with a plant, you see in one, picture here. There is more to this. I will come back to that later. So here we had a direct contact assay. So we applied the microbes directly to the plant surface. But you can also look at the impact of microbiomes, from a distance on plant growth. And the system that we developed, another PhD student in the lab, so this is a pot filled with soil and a plant. And then you have a membrane here, and underneath the membrane there is an airspace. And then at the bottom here you can put a soil, you can add a microbiome, you can eliminate a microbiome, you can even put a Petri plate with a single microbe there. And the idea is that the microbes here, due to their activity, they start to produce volatile compounds, which then impact on your plant growth. And that's what you see here in this picture. So the sterile control here, and then a plate where these are lettuce plants, where at the bottom here you have a soil with an active microbiome, right? Again, here, if you do this assay, you can already, also with the previous one, you can have multiple phenotypes as an outcome. So again, once you have this phenotype, you can do all the tools that you had learned in the last week, right? So you can do taxonomic analysis, you can do metagenome, which will be explained this afternoon, and then you can basically disassemble the microbiome in this soil, start pinpointing microbes that are differential depending on the treatment that you have, and then start to build these microbes one-on-one, or build a synthetic community if you like. And then you can test that again under very controlled conditions in a plate assay here, where you have a little plate here, and then you have your plants here. You see, and you can see that this microbe here gives, again, a different phenotype on the plant, but not in a direct contact assay, but from a distance. Okay, good. So coming back to this picture, there is no blind date without chemistry, right? So what Jason did is he also looked at the chemistry. So what he did is he took the leaves of these plants. So again, the microbes are on the roots, not on the leaves, but he took the leaves, just looking at how do these microbes impact on chemistry. And the reason why, and that's, I think it's a growing, interesting field of plant microbiome research, because microbes can also change, the plant chemistry, especially of plant secondary metabolites. And there are quite a bit of plant secondary metabolites which we know well. So the coffee, the glucosinolates here in the broccoli, and also in wine you have some interesting compounds that can also have a health beneficial effect. And then of course we have the beans, the artemisinin I just talked about. Yeah, and this is, Jason put this one in. That's also a plant secondary metabolite which is used by humans. So in a review in 2018, we listed a number of what they call HVNPs, high value natural plant products. And this is basically, you can classify them in different chemical groups. And these is just a snapshot of what we put in that paper on all the different plant products. And the idea of this experiment was, again, to see if we apply a single microbe or a microbiome to the roots of plants, how does it change, shoot chemistry? And the experimental work that Jason did, so these are the plants growing on the plates. So you add the microbes, right? You let the plants grow, and then you cut, You put the leaves in a mortar. You add some liquid nitrogen. You grind it up. And then, it's ready, with some additional extractions to do mass spectrometry or another type of analysis in terms of chemistry that you are interested in. And then you go through, what is quite interesting, you go through a pipeline which is not so different than you're using here for microbiome analysis, right? So there's quality checks, etc. There's the clustering, there's the PCOA analysis, and finally, compound identification. At the same time, Jason also did the phenotypic measurements of these plants, and then at some point you can also relate chemistry to the phenotypic readout. And this was the result. It's not so much so, this was a lot of work where you actually have to classify each of the compounds that came out of the analysis. And of course, there's a number of unknown compounds in there, but also a number of known compounds. So we were interested, for example, what happens here in this too much love can kill you phenotype, right? We expected that the anthocyanins, which gives this discoloration of the shoot, should be there. And indeed, they were much higher in this microbe-3 combination, but also other compounds like camelexin and salicylic acid, which is a plant hormone, were also upregulated here. So defense systems were way up in this combination, although it did not really help. Then going to these high-value natural plant products, of course, this phenotype here on the Artemisia. And what we found out, so you have to normalize, again, peak intensity per biomass. But even if we normalize in this third combination here, you could see that the levels of this high-value natural plant product were way up when you apply a microbe. So think about it. There's plant breeders. They like to also do breeding on these types of plants to get much higher levels of this natural plant product. This told me that you can also, without genetic modification of your host plant, you can also use microbes to trigger or to enhance levels of specific medicinal properties of plants. And there's a lot of groups worldwide that work on the impact of microbiome on medicinal plants. Are there people here working on medicinal plants and microbes, interactions? No? Okay. Victor, you? Is it on? I think it's dead. It's blinking. Can you still hear me? Or shall I stand here? Okay, good. This is better, right? But tell me if my voice is too low. Okay. The phenotypes that I've shown in the experiment show this nicely branching, etc. There are a number of groups also working on microbiomes and root development. And then both on the microbe side, but also on the plant side, you have beautiful tools to visualize specific physiological processes. Like here, for example, they used an auxin reporter, to visualize changes in auxin levels in the roots related to root development. And then also groups, and I'm not going to tell too much about it because there's one paper in the literature that you are going to present, which talks about the impact of microbiomes on shoot development and also on flowering time, right? So, and that is depicted here. So root microbes that have an impact on above ground development. And there are some nice relationships with, again, hormones, indole acetic acid, but also the nitrogen cycle plays a role in that phenomenon that you see here. I'm not sure, no, this is not the paper that is in the literature, but there is a really nice paper where they actually managed to identify microbiomes, so-called early flowering microbiomes and late flowering microbiomes that indeed, as the word says, has an impact on the timing of flowering of these plant species. A control here, this was the sterilized soil where there was a residual microbiome still present, but still you could see that the early flowering and the late flowering, they separate nicely from each other. And they even did some transplant studies in this study where they transplant early and late flowering microbiomes to a whole series of genotypes of this plant species and see how do these genotypes respond to these microbiomes. Okay, so the last microbiome-associated phenotype is about stress. There's a number of stresses here: biotic stress, nutrient stress. There's here thermal stress or even freezing, and then of course the usual suspects here, drought and flooding. In the outer ring, there's a number of traits within the microbiome that contribute to the tolerance to these stresses in the inner circle. So have a look when we share the slides. I'm not going to go through every little detail here. So again, this is, these were the phenotypes. Now let's go into disassembly and reconstitution. Because how do you do that? So you have a microbiome, you have a phenotype, you establish a collection of bacteria, fungi, or other microbes, and then how do you reconstitute these microbes? Yeah. So the first is that many groups, including my group, we start testing one microbe at a time, right, to see if they give a particular phenotype. But at some point, you also want to reconstitute the microbiome as it was originally there. And there are many different rules. You can say, let's reconstitute the microbiome based on differential abundance. You have learned about how to establish differential abundance, right, between two contrasting situations. So that's a way to build your community. So you take different taxa that are differentially abundant, you test them first individually, and then you start to build the community based on this. You can also reconstitute not only based on taxonomy, but also on functional diversity in terms of differential abundance. And Victor and Luisa will talk about the metagenomics and differential abundance on metagenomic traits this afternoon. Another way to build or reconstitute a microbiome is using network analysis. You will see papers where they do a lot of what they call co-occurrence networks. So when one microbe goes up and the other goes up, there's a positive relation, but sometimes also there's one going up and one going down. So the structure of these networks, you can also use it to design your consortium or your synthetic community of microbes. And there are ways, there is also, so you can first make a complex community based on the network, and then you can also well, in the network analysis, you can also identify so-called hub taxa. This is a hub, like in a metro, right? So there's a central point where many, connections come together. So these are quite important because if you, let's say, drop them out, so if you take them out of the consortium, then you get a disconnected network. Whereas if they are not essential, the network will stay intact. And the last, way to do this is to reconstitute microbiomes based on functional traits, right? And that type of information you can infer from the metagenomic analysis, and also from experiments where you test individual microbes for a particular phenotype, and then at some point you bring those together and see if, when you bring them together, you get even a better phenotypic readout. Are these rules of the game clear? Right? So, yeah, so keep in mind that there are some, ways to design a synthetic community or a consortium. It's not like, okay, let's just put them together and pray, for a beautiful outcome. Okay, so I'm going to give you an example on disease-suppressive soils, which I think is a very nice example where you disassemble a community and then reconstitute them into the phenotype. Are people familiar with the phenomenon of disease-suppressive soils? So these are soils where a plant does not get diseased or only little, even if the pathogen is present and the host plant is susceptible to the disease. Yeah, so that's the key definition here. And it's been described for fungi, bacteria, nematodes, and even for parasitic weeds. It's microbiological or biological in nature because if you do a heat treatment, you remember the experiments that we said. So if you do a heat treatment, you lose the effect. And when you transplant, so you mix suppressive soils with a non-suppressive soil, a conducive soil, you transplant the level of suppressiveness. The suppressive soils that we work on is suppressive soils for a fungal pathogen, Rhizoctonia solani. Rhizoctonia solani you also used in your practicals here. Yes, you had 2 fungal pathogens. This is one of them. This pathogen is also super important here on rice. It's, I think, Kumar, if correct me, it's in the sheet blight. Yeah, it's in the top 3, I think, right? That's another sort of like what they call anastomosis group than the one we are working on. So there is some host specificity here. The one we are working on is on sugar beet. But I'll just show you always when you start working on pathogens and diseases, you always look first at the life cycle. So how does this pathogen infect plant tissue, and how does it survive inside the environment outside the host? And this is a little movie that was made by APS, and I'm just going to show you. This is not so interesting, but as soon as it goes underground, you will see that this pathogen survives in soils through very resistant structures, so this is the hyphal network. And when the hyphal network clusters, it forms these survival structures, these ones, which are called sclerosia. So sclerosia, they are quite resistant to, adverse abiotic conditions. And when these sclerosia, when they receive a signal from the host plant, they germinate. So these are the root exudates. Again, they respond to the host plant in terms of root exudates. And then they grow towards the plant root. And once they reach the plant root, they colonize the surface through these what they call runner hyphae. And the runner hyphae, they cluster into a cushion. And from the cushion, they start to produce cell wall degrading enzymes. And then the fungus penetrates the plant tissue. So it's a necrotrophic pathogen, so it destroys the plant from the outside. Then it grows inside, and you see discoloration of your roots there. And of course, the whole, it's destroyed these cells. and the plants, they either are infected at the early stage, so they call this pre-emergence damping off, or they are infected at the later stage when they first emerge, and then it's called post-emergence damping off, right? So that's the disease cycle. The interesting is that, and you can see, again, these discolorations of the roots. You see the root tips. They have this spear tip, a typical symptom of Rhizoctonia on wheat and also on sugar beet. And then in the field, you see these empty patches, usually, which typically is related to either pre- or post-emergence damping off. Okay. So the interesting part of suppressive soils is that in the field, so it's a field phenomenon, so they develop in the field in this type of dynamics, and not only for Rhizoctonia, but also for other fungal pathogens. So here on the x-axis, you have the time of your cultivation of your plant, so multiple years, and here you have the disease severity. So in the beginning, when you start growing a specific plant species in that field, there's no problem. And then at some point, you get some a buildup of your fungal pathogen, but the initial root infections, they are still suppressed here through basal defense mechanisms in the plant. At some point, this is not effective enough, and you get a severe disease outbreak, usually after a number of years. Yeah, the typical classic. And then many farmers at this point, or even a bit earlier, they abandon growing that plant species and they start to grow other plant species. But if you continue, and that has been shown for a number of fungal plant pathogens, you get in sort of like what they call a decline, disease decline. So the pathogen is still present here, the host is still susceptible, but you almost do not get disease. So here the plants, they enter in a suppressive state. And that will continue as long as you keep on growing the host plant, right, in that field. It's interesting, if you take soils from this point here and you introduce the pathogen at a high density, you do see a slight increase in disease, but that's quickly suppressed again, and then it goes back into that suppressive state. And this is, again, the phenotype for the system that we work on. And we showed that already many times in the previous lectures, right? So this is how the plants look like in the suppressive state. Pathogen is still there, host is susceptible, but there's no disease. So we were interested in, okay, what is the difference between those two? And again, we went through the different types of analysis that we also discussed, earlier last week. We did amplicon analysis, metagenomics, and then we started to disassemble the community by plating and then reconstitute basically this community. And the readout here, the phenotypic readout, was disease suppression. Okay? So actually, when my kids were very small, I always explained my work with this cartoon. And with some lectures at home, I leave this picture on, and then I leave the classroom, and then I come back, and then I hope somebody has seen the difference. But when you do this type of analysis in terms of microbiome, this is actually what we are doing, right? Which you have been doing last week also when it comes to taxonomy. So we have the disease-conducive soil and the healthy soil here. So if you look at the analysis that we have done, so the two contrasting situations, in fact, we do not see a real difference in composition. But we do see a clear difference in differential abundance. So the abundance of specific groups goes up, and from others it goes down. And that gives rise to that particular phenotype. And that is shown here. So this is actually a pie chart of the differential abundant bacterial taxa. We focused on bacteria, but we could have also focused on fungi here, or maybe even another group of organisms. But our prime focus was on bacteria. And then Victor also showed this picture in his earlier presentations. So we have, basically the collection of the differential abundant taxa, and they are listed here. And then actually the tedious work starts. So you gotta isolate the microbes that were pinpointed in your amplicon or metagenome analysis. and then try to match, like we also have shown in the, basically the bioinformatics flowchart. You have to match your culture collection, if you can culture them, with, your differential abundance analysis. And we have done that for gamma proteobacteria, beta, planktomycetes, firmicutes, and actinobacteria. And I'm just going to show you a little example, not from all of them. So again, this is the targets, and you start isolating them. You purify the colonies. You have also done that in the practicals. You profile them here, and you do the amplicon analysis to match your collection with the amplicon data, or to match your functional genes with the metagenome data. And then those isolates which then match, you test them in this, Victor talked about this sausage assay. So here the microbes are added. You add your fungal pathogen Rhizoctonia here, and then it starts eating its way through the row of plants. And because it is a post-emergent damping off disease, your plants fall over when they are infected. So you can actually measure the distance that this fungal pathogen is traveling through your row. And if it travels all the way there, it means that your bacterium does not contribute. If it stays close here and those seedlings survive, it means that you have a positive readout in phenotype. Those microbes you can then also test in plate assays, which you have also done in the practicals. And those bioactive species and strains you can then do genomics, mutagenesis, and even chemistry to see, what are the genes, what are the metabolites involved. So we have done that for Pseudomonas and Lysobacter. Victor also showed you Burkholderia, right, with the volatile compounds. And this was the work of Pseudomonas, where we discovered a new antifungal metabolite, which we called tannomycin. And tannomycin, that's the product of a non-ribosomal peptide synthetase pathway. And it's quite a complex structure here with a fatty acid tail linked to a cyclic peptide. So this was a really nice example. So Pseudomonas was differentially abundant, and indeed we could confirm with the phenotypic readout that when you reconstitute it, that it does contribute to the phenotype, and we actually discovered a new compound here. So we did the same for Lysobacter. So there were two papers here, basically the same workflow. So we looked at the diversity and activity. We did comparative genomics and metabolic profiling. And the end result was that this genus had super interesting in vitro activities and also genomically very interesting, but it did not give the phenotypic readout that we were looking for. So that's also well possible. Then we started to combine this Lysobacter with some of the others, because maybe Lysobacter by itself does not give the effect, but only when it's in a consortium. Also there it was of no added value. So keep in mind that the differential abundance type analysis gives you a first hint or a clue of what to look for, but the validation, so the experimental validation, really will tell you whether it's functionally in that system or not. Okay? So, and then this is actually the cartoon where we have, this is the root, this is the rhizosphere, and these were the mechanisms in the rhizosphere that we have discovered so far. So we talked about Pseudomonas with this cyclic lipopeptide. We talked about the Paraburkholderia with some of these volatile compounds. And also for two actinobacteria, Microbacterium and Streptomyces, we could show that the activity that they contribute to that phenotype producing these volatile compounds. One thing we are still working on is this story here, because we also see an enrichment of oxalobacteria and Rhizoctonia, but also many other plant pathogenic fungi. They use oxalic acid as a virulence trait in infection. And we suspect, but we have no experimental proof yet, that some of these bacteria, they utilize the oxalic acid as a carbon source, and thereby they basically diminish the virulence of the pathogen. So the endosphere, I'm looking at Victor. There's quite some metagenomics done here and also transcriptomics. Let's see this afternoon if there's time, to go through this, the endosphere. So when the pathogen is able to break through this first line of defense here, and it enters the root system, can these endophytes also give an extra protection? That's basically the topic. Okay, so disassembly and reconstitution. Again, one of the figures from the review by Julia Vorholt here. So you can have what they call low complexity communities, and you can also have high complexity communities. There are papers out there where they reconstitute microbiomes of more than 100, 200, microbial species or even strains within a given species. We tend to, keep it still at a low complexity, at least in my lab, because for me, handling one is already complicated. Two is, anyway, so we try to stay at the low complexity level here. The GM space here, so the advantage of this, there's a high reproducibility, and you can much more easily establish causality, which is an important element here. The risk, indeed, is that you're missing keystone species or hub taxa. Here, reproducibility is high, but it's much more complex to establish causality. GM stands for the genotype of the microbiome. So what, the functional traits that the microbiome contributes to that phenotype. Okay, so let's give a few examples of the low complexity interactions, and that's here. So I listed a few interactions in terms of mechanistic things like competition, anabiosis, interference with quorum sensing. There's somebody here working also on quorum sensing. There's some nice work also done where microbes produce an enzyme that basically degrades the acyl homoserine lactones, so the quorum sensing molecules. But I want to go to an example of post-harvest diseases. Are there students here working on post-harvest diseases? I think there are, right? Okay. I heard somebody's working on aflatoxins, right? Yeah. Typical also in post-harvest, diseases. Banana as well? Okay. Yeah. So this is sort of like a very classic example. I think this is a penicillium, indeed, of oranges. So they did an experiment here where they damaged the skin of the oranges, then inoculated, non-inoculated or inoculated with Pichia, and Pichia is a yeast. And then you can see this US7, which is the Pichia, you get beautiful protection. And especially with post-harvest diseases, the risk assessment analysis is super important because you're basically applying microbes to an edible product, right? So also for this one, they had to go through a number of risk assessment studies, and basically in these risk assessment studies, they want to know if the microbe does not produce some sort of a compound that can be toxic for the persons that take these products in. And for the yeast, they could not discover actually a specific active compound produced, and they suspected that this yeast protects the oranges by colonizing these, these damaged tissues and eating the substrates, providing that the pathogen can enter through here. A similar post-harvest disease, monolinia here on, I think it's peach, and then we have a bacillus treatment and we have a fungicide treatment, right? So which of these two would you prefer to eat? So this is Benomil, fungicide treatment, bacillus treatment. Any preference? Sorry? The bacillus, you all want to eat the bacillus, okay? Good. So they did some chemistry on this bacillus. And here is the HPLC profile. Here you have the bacillus and the monolinea around it. And these are all the chemicals that are produced by the bacillus. So again, emphasizing that although you have the feeling like, well, it's a biological, so it should be fine. Again, there's always chemistry here. So you need to do, in terms of risk assessment, you really need to do profiling of the chemistry of the bacillus when it's applied to these types of edible products. In terms of the mechanism, so once you have an interesting microbe and you want to identify, which compound is actually responsible for the activity that you see, there is a number of approaches. The first thing, of course, is you have to determine the activity of the bacterial strain, and there's a number of assays in vitro that you could do. Although, keep in mind, like Victor said, he did these assays with the Paraburkholderia and he could not identify any activity until he did the sandwich assay. And then he discovered there was a volatile activity. So then you isolate you isolate the active compound, you resolve the structure, and then you compare the activity of the compound versus the bacterium. That is basically more like the chemical approach to look into the mechanistic basis of the interaction. I know, Kumar, that many of the students here, they do extractions, right, on plates or on liquid cultures. There is a technique that we used in one of the projects together with an American group in San Diego. And it was a technique where you could look at the chemistry live. So you have the agar plate with your microbe of interest or in an interaction. And then with this needle here, you can deliver a solvent at that point where you are interested to look at the chemistry, and then the solvent goes into the mass spectrometer, and then you get an immediate readout. Then you can put the plate back into the incubator, and the next day or an hour later, you can do the chemistry again. And Peter Dorestein, he developed this technique. It's called nanodesi. And we also, in our paper, we also have this little movie, so you can download this also, where the technique is shown. So again, here you have, I think this is where the droplet is delivered on the bacterial colony, and then it goes into the mass spectrometer. So just to show you how it works, so it goes down to the place where you want to look at the chemistry, and then it reaches the contact, and then you see this little droplet that is delivered, and then on the right you can already see the readout in the mass spectrometer. And then, with a database there, you can then assign even compounds to the masses that you find in the readout. Yeah, and then you basically lift up the system, and you put this one back into the incubator, and then, so you can look at chemistry in a dynamic fashion. It's not let's say, destructive sampling. And many labs, you can build this actually quite easily yourself. So coming back, so in several systems they have done that. So with the Burkholderia, they identify the compound. So this is the bacterial cells, and this was the compound, the AFC, the antifungal compound. And when you apply this to the seed, you could indeed prevent this damping of disease on cotton. And then the other approach is not so much the chemical approach, but it's the approach on the microbial side to identify the genes. Again, you start with the activity, and then you can do a mutagenesis, either random or site-directed. And site-directed, you can do that if you already have knowledge, based on your genome information, which gene clusters might encode for specific active compounds. If you have no idea, you have to go through a random mutagenesis and then try to find, the genes that are involved in the activity. And then once you have the mutants, you compare the activity with the mutants and the wild type. And then usually what they also do is you do a complementation of the mutant. So you bring back an intact copy of the gene back into the mutant, and then that should restore your phenotype. in situ expression. So are these genes actually expressed in the real system? And you can then also even express the genes in another host. And that's called heterologous expression. And then can you then confer activity? Just some examples here. Fusarium on chickpea, nematodes on potato, and these are some of the bacterial strains here that give the activity. So this is the activity, healthy plants on the y-axis, and 2 mutants, they have lost the activity. Here, this is juvenile mobility of the nematodes, high mobility in the control. It's suppressed in the wild-type treatment. A mutant loses it, and when you complement the plus, it's back. So that's usually also, thinking about mechanistic studies, also in terms of publishing and journals. This is usually, when you go to validation, what they would like to see. And then coming to the last point, so are these genes that you identified, are they actually expressed on a root, on a leaf? Yeah, and for that you can use reporter genes. For example, the GFP, where you show that the expression of that gene is actually also the case in the real life situation on plant leaves. Because remember, also from the practicals, sometimes you can have beautiful activities on an agar plate, and those microbes will show no activity when you go to a plant. And that can have to do with the expression of the genes in situ. This is an example. I'm not sure if we put this paper in the literature discussion. I don't think so. But this is an example of antibiosis also in the phylosphere. I know a number of people here are working on the phylosphere. And this also talks about all these 2 different types of approaches, using chemistry, but also using mutant approaches to identify, the interactions that occur on the leaf. One more example on low complexity interactions is the induced resistance response. Are there students here working on induced resistance? So where the microbe triggers a resistance response in the plant, which then suppresses the disease. Nobody? Well, maybe you are working on induced resistance and you're not aware of it. That's well possible. Shanu? Yeah? Okay, good. There is a number of research groups. This is working on induced resistance. This is a cartoon from the group at Utrecht University of Corné Pieters's group. The nice thing of working in terms of functional analysis of the induced systemic resistance is that they make use, of course, on the microbe side here, which traits determine the activation of the immune response. But working with plants, and especially with Arabidopsis, you also have access to a number of mutant lines in Arabidopsis, which you can use to find out how do these microbes actually trigger the induced resistance response. And that is shown here. So in Utrecht, what they discovered is that specific microbes, they trigger an induced resistance response through a gene called MIP72. And MIP72, that's a transcriptional regulator. So that basically activates the resistance response. And recently, they discovered that there's a specific hydroxylase enzyme that starts, the biosynthesis of a coumarin. And a coumarin, and the coumarin that's scopoletin. And there's one enzymatic step more, which is a glucosidase, which then transfers scopolamine, that's the metabolite inside the tissue, to scopoletin, which is then secreted. The interesting of scopoletin is that they have shown in vitro that it inhibits specific microbes, especially pathogens, and it favors, the growth of beneficial microbes. So there is a differential activity of the coumarin. I know that they are working on this more to see how broad is this selective effect, inhibitory effect on microbes. So this is a nice example of, basically an indirect way how microbes can, in a low complexity system, prevent the infection of plant pathogens. Okay, what about unknown mechanisms? Suppose that you are working on a system where you have no clue about the mechanisms that is operating, right? I'm going to give you an example of a mechanism that we discovered without knowing it when we were trying to control so-called zoosporic pathogens. So we're talking about Pythium, Phytophthora, those types of pathogens. And again, there is this APS Press YouTube movie about the life cycle. That's always where it starts. And I turned off the sound. So this is an oospore. That is the survival structure of a zoosporic pathogen. And it forms a germ tube. And then on the outside, you see this vesicle. So that is here. This is the vesicle. And in that vesicle, there are some motile spores formed. And they have flagella. And these flagella, especially when the soil is wet, they allow these pathogens to spread over quite a distance. So they are released from the vesicle. And again, they are chemotactically attracted to the exudates from the plant roots. So they go to the roots, and what is super interesting is as soon as they touch a surface, they lose the flagella, and they round off, and they form a cyst. And this cyst also protects them against some abiotic stress. And then the cysts, again, germinate, following, again, a signal on the host, and then forms an appressorium, which then, again, gives a penetration hyphae using cell wall degrading enzymes, like I've shown also with the Rhizoctonia, and then it also penetrates the tissue. And again, because of the activity of these cell wall degrading enzymes, you get disintegration of the plant tissue. And like with Rhizoctonia, although this is not an actual fungus, but it's an oomycete, you get sort of also a damping off disease. And because of the damaged tissue, these plants also become much more sensitive to other pathogenic microbes. So knowing about the life cycle, what we did in our group is we said, okay, so one of the key parts in the life cycle is this zoospore, right? So they move from the vesicle to the plant. So let's target microbes that do something with these zoospores. So what we did, super simple experiment, we generated in the lab, we generated a culture of the Phytophthora in this case, the late blight disease, and we generated the zoospores, and then we had a huge bacterial collection, and we started to screen all these bacteria for activity against these zoospores. And there were a few bacteria, so this is the life cycle here again. This is a microscopic movie. So this is the zoospore, and the little guys around it are the bacteria. And this is the phenotype that we saw under the microscope. Right? So that was the phenotypic readout that we had with this bacterium. And then we went through the two approaches that I just mentioned. So the first one, is this an active compound or not? So, and we went through the chemistry, and the active compound that we detected here was this compound, again, a lipopeptide, fatty acid tail, and a cyclic peptide. So we could mimic this phenotype with this compound. And then, like we just said, we took the second approach. We were interested in the microbe, which genes and regulation is part in the production of this compound. So, and because at that time we did not have any information on the genomics of this strain, we did a random mutagenesis. And for that, just to show you here, we screened 7,500 random mutants for this particular phenotype, and we found 55 mutants that did not show the explosion of the zoospores. And then we sequenced the genes around the mutation, and 40 of them were in so-called non-ribosomal peptide synthetase genes, right? These very complex genes, and the product of that is not synthesized by ribosomes, but by a peptide with all these different domains, right? Okay, so that was basically here the low complexity part. There are a number of papers on high complexity interactions, and I list two of them here. One from Julia Voorholt's group on the leaf microbiota, and then here, I think everybody may be familiar with this paper, but this comes from Kumar's lab here, where you looked at the phylo microbiome of rice and then against blast disease. So I think that's also a beautiful example of high complexity interactions between microbiota and pathogens. Okay, so this is the last summary slide. I think we're nice in time. Again, showing the initial slide of Julia Voorholt with the disassembly, reconstitution, phenotypic readout. That is also shown here, but this is a bit more detailed. So this is the plant system. You get the deconstruction or the disassembly where you establish a culture collection. You can characterize the networks and identify the hub taxa, the core taxa, reconstitute these and screen for specific traits here, and then at the end design a so-called synthetic community which you can then test again back on the plant. Okay, I will stop here. I can see this may be a lot of information. Are there questions about this? Are there things not clear? Yeah, please. Do you have a microphone? Well, first let me ask, was the story clear? Right? So keep in mind, deconstruction, reconstitution, disassembly, reconstitution, that's super important. And then the different ways to, construct these communities, yeah, based on abundance, function, networks, right? Please. You mean what would be the best strategy to look at? I don't know. I don't know. So we usually go for reconstitution based on function, but I know there's also groups going for reconstitution based on networks, because many times with functions you have to have a readout, right? But some taxa, they don't give a phenotypic readout, but they are super important for stabilizing the community. And then the whole network analysis is going to be crucial. And one thing I have not even talked about is how do you actually reconstitute in terms of density? I mean, that's a super difficult. So many labs, they reconstitute in a 1:1:1:1:1 ratio. But if you go to basically the microbiome analysis of your system, these microbes that you pinpointed as being important for the phenotype, they are not there in a 1:1:1:1 ratio. They are different. So some labs, they say, yeah, but if we apply them in a 1:1 ratio to the plant, they will very quickly sort of like settle in the ratio that they normally occur, which they have also shown in some work. So because imagine the matrix, right? So you have 1, 2, 3, up to 10, 15 microbes, and you have to make combinations of those. That's already massive. And if you then even have to take into account the density of these microbes in this community, I think you need, 20 greenhouses to do the experiment. So it's a challenge, yeah. Yeah, yeah, very good point. I did not mention that, and that's the importance of what they call tractability. Yeah, so if you have a small community with, let's say, 5 or 10 members, can you, after you apply them, can you track them and specifically track them? There are ways to do that, to label them, each with an individual label or a marker. Or sometimes they have by themselves already very specific phenotypic traits that you can monitor them by plating. Yeah, tractability there is the most important. If they go in, they stay out, the density, dynamics, yeah. Any other urgent questions? Shannon.
Model plants. I would like to know the composition of the media which we use for the experiment. I'm guessing, is it mineral soil medium which we use for it?
Right, it was, so that was a half times MS medium. It's a Japanese name, Mushkari, something like that. Yeah, half times MS, yeah. And the medium, which is quite nice, it's very good for plant growth, not so much for microbial growth. So the microbes, when you apply them, they don't start to, grow into these huge, they stay super, and they only grow because of the exudates that are released by the plant. Yeah, so the composition, I think you can, you will probably find it, Shannon. Yeah, okay.
Mainly we are using bacteria. Like, can we create a synthetic community with fungi as well as bacteria? Like we have fungi like trichoderma.
Yes, yeah. There is nice work from the Max Planck Institute in Cologne, where they started to design synthetic communities consisting of both fungi and bacteria. Yeah, Stefan Hackar, that's the name. Yeah. With fungi, it's since they are not single-celled, yeah, it's a bit difficult to make a community, but with trichoderma, for example, you can use the spores, right, to reconstitute a community. With some other fungi that do not form spores, you have to find a way to make the inoculum, for example, with mycelium, right? But yes, there are several examples where they made communities with fungi and with fungi plus bacteria. Yeah. Yeah. Are you good? Monday morning. Okay. Yeah, in the back. Network analysis, Victor.
Co-occurrence. Okay. So co-occurrence network. So what you do basically to generate those networks is to correlate the presence and absence. Yeah. All the different ASBs or ODUs present in your environment, in your sample, and then this is going to give you a value, yeah, that you can transform into a correlation. Well, you have different types of correlation. You have sequence correlation, you have quite a lot of tools that you can use to generate these correlations. I can recommend you some. For example, we use a lot of SPACC, and SPACC there is a tutorial in GitHub that you can follow to generate. It's not that difficult to generate. If you have yeah, actually you need a server also, because imagine that you have a huge table, yeah, with a lot of OTUs and ASVs, and then you have to generate correlations one to each other.
Yeah.
That gives you a long list. So then you need some computational power there. Yeah, I can recommend you later in the afternoon where we are doing the identity network. I can recommend you some tools for co-occurrence.
Yeah.
Yeah. Does it help? Please? You mean co-occurrence? Yeah.
Victor, can you repeat the question?
So the question is that if we can do the same like co-occurrence networks in metatranscriptons and maybe also metagenes.
Yeah.
The answer is yes. So you can do it. You can do instead of co-occurrence, you do co-expression networks for the transcriptome. Yes, exactly. So you can do that for any kind of data. So even for genomics, you can do also kind of network analysis, DMTS networking. So, but for the transcriptome and metatranscriptome, you use other parameters. I'm not very familiar with it, but I know that there are some tools that can do it for you. Maybe Guillermo, you have done that. At least in a genome, that's transcriptome. Yeah. Yeah, you can do a expression network. So basically, you have your node. So imagine that each node is one gene, and just in the transcriptome, you can have one taxa or OTU, and then they are linked together of how their expression changes in your experiment. So if two genes when you are doing the culture in your experiment, having a similar trend in expression, they will be together. So maybe they are linked one to each other, or they are involved in a similar function. So that's helpful to also, for example, if you have one specific gene that you know that is interesting for you, you can see which other genes also like evolve or express in a similar pattern. So they are associated. So you will also do, let's say, extend the mechanism that you're interested in.
So I was asking, here also the coordination will be the let's say on our base of coordination only we will be looking at transcript analysis on the residual coordinate. First we will be finding the coordination of them and then we will be collecting together. That's it.
It's not that basic correlation, the co-expression ones, but you can understand the similar. Like you calculate the distance between each gene, and then the distance is based on how they evolve in your experiment. So for example, you have one gene that is in one condition one, in the condition two, in the condition is value of 3, then it will find you other genes that are also having the evolving 1, 2, 3. But it's not based in correlation. The nature is kind of the same. So, for example, imagine that you have a group of genes that are all expressed at the same time and more or less at the same level. So when you more taking into account expression levels. Okay? And the other type of networks is the identity networks, or the similarity networks, where you take gene by gene and then you compare, and then you get an identity, like 97% identical or not at all, like 40, 50, 30. And then you can also plot that in an edge. If there is time this afternoon, I will show an example. It can go on because sometimes you represent this distance in another way. For example, you remember when I was explaining the Jaccard index? You can use also Jaccard index to represent the distance between nodes. And that is showing you the dissimilarity between nodes or genes. Okay? So I will show an example this afternoon. I hope there is time.
Yeah, Vic, that relates to the end-of-fight work, right, that you have done, right, to see if certain functions in terms of identity are unique or whether they are part of a pool, right? Yeah.
This afternoon I will try to expand a bit more on that.
Okay, Ramakrishnan, I'm looking at the time. Is there?
We can reach by 10:45 now.
Okay.
No more questions.
That we have the whole day, so if there are other questions that come up following the lecture, we have the whole day, right? So just ask.
This is the only day where you can interact with people personally. So make use of it. I said this is the only day where they can interact with me personally.
Yeah, personally, yeah. Well, I mean, later on, if things come up, we will also provide the email addresses from us. You can always contact us by email, okay? Yeah.
Okay. Now it's time for us to move to the auditorium in plant technology division. So we'll be there by